Let's talk about engineering synthetic life. This technology has been here for a while, and yes, we as humanity have made living things from non-living things. There's artificial bacteria, there's artificial yeast now. They're trying to make an artificial human genome that is probably never going to be cloned into a person, but yeah, it's probably possible. Blade Runner, anyone? This technology, however, has a good amount of flaws, and as we are moving toward using it in more medical research and medical treatments, it's worthy to consider what maybe we can fix and what could go wrong. So typically, when we engineer something for technology, it involves taking a gene that doesn't belong in a bacteria and putting it in. It can also be done with plants, so you can make new crops. However, you might think that it's a very complicated system, and it is. With top-down synthesis, we're essentially hoping that everything goes just right, and maybe it will, but under certain conditions, it could crosstalk with other genes and go wildly out of whack. And you do not want that to happen inside of a human body if you're using that microbe as a treatment. By the way, for the large part, phages have been used for gene editing in humans when we're talking medical treatment, but there are other applications, particularly with biohybrids. Aside the point, if you are just engineering one gene, you are not fully aware of all the circumstances in which that gene could be active or what it might introduce that could interact with other genes. This is why it's probably better if you are going to use synthetic biology to work bottom up rather than top down. Meaning you assemble the genome, you know everything that's there, and hopefully you understand it well. Minimal genome, by the way, is a field that I've worked in with microbes, trying to figure out the smallest possible genome you need to be alive, which goes into making synthetic life. We as humanity first built Mycoplasma laboratorium, the very first synthetic bacteria, and since then we've built yeast. Yes, synthetic eukaryotes like ourselves. I don't know how to say this, but building a eukaryote is alarmingly complicated. We're talking about many orders of magnitude more complex than a bacteria, but yes, it can be done. And of course, there is an international effort to make a synthetic human genome. So this would be every gene that a human would need to, be a human put into a cell, grown in a lab. And this is not for cloning people, although it probably is possible. This is to understand our genome and solve the problems that we have in synthetic biology. If we can build a human genome from the ground up, that means that we can understand every single gene, how it interacts with each other. It would make an unprecedented study of what exactly our genes are doing, which are not entirely understood. This is a massive leap forward, and of course we are not there yet. Now, the other reason why it is important to do this is because we're also making biohybrids now. So electronics that interact with bacteria or other organisms, often used in medicine. Of course, it could be used in industry because it turns out borrowing these sensors from a body, like a sense of smell from a moth, as it would turn out, is actually pretty efficient for robotics. If we are to make electronics that hybridize with biotic stuff, it's going to be a lot easier to engineer the biology to do so, rather than just trying to hijack it. And hijacking is fine and good, but there's more efficient ways. Think the tiny lab-grown brains, except they are engineered to do just computation. It's going to be quite a bit more efficient, I hope. If I could impart some knowledge, yes, I understand this sounds really spooky, because it's also really new. We're not used to it. I mean, I'm pretty sure if someone gave you the concept of factory farming, it would have been horrifying a few hundred years ago. It is still horrifying now, but we all kind of exist alongside it. I mean, the idea of taking fungal byproducts to treat infections is a little bit spooky too. This is an up-and-coming field. There's a lot of good that has been done and will be done. I mean, insulin is now made by E. coli who have genes to produce insulin, so a lot of it has already existed. I don't know if I am or am not the right person to say that it's looking pretty optimistic right now. If there is a problem, I will let you know, but nothing to worry about for now. When the robots rebel, I will tell you about it. Follow for more.